day everyone, welcome to the first class of the course Next Generation Sequencing Technologies, Data Analysis and Applications. In this class, we will introduce some of the methods that were used before the next generation sequencing technologies came about. This will help you understand how next generation sequencing technologies have impacted the fields of genomics, transcriptomics, metagenomics and epigenomics. And following that, we will also introduce what is next generation sequencing, and in subsequent classes we will talk about these technologies one after another. So, let us begin. This is the agenda for today's class. So, we will start with Sanger sequencing method. So, this is the first direct sequencing method that was available. We will talk about microarray technology. This is a high throughput method that actually allowed some degree of mutation identification and gene expression analysis. Then we will introduce next generation sequencing, the idea of next generation sequencing. We will not go into the methods in this class, and then finally, we will talk about some applications, so that you understand how these methods, next generation sequencing methods, they have actually impacted our lives. Okay, so, let us start with Sanger sequencing. So, these are the keywords we will come across: dideoxy termination reaction, sequencing by synthesis, DNA probes and high throughput sequencing. So, Sanger sequencing, this is the first direct sequencing method that was proposed in 1977, and for that there was a Nobel Prize because this method had so much impact, which allowed us to actually first look at the bases of DNA molecules, right. So, we could read these bases now one by one. Okay. So, how does this method work? So, this is a sequencing by synthesis method, and that means we need to synthesize the complementary strand of a molecule that we want to sequence, right. So, imagine I give you a DNA molecule and we say let us find out the sequence of that molecule. So, what we have to do is we have to synthesize the complementary strand of that molecule, right. So, for that we need polymerase DNTPs, right. You know what kind of process we need to do, right. So, we need these molecules to actually synthesize the complementary strand. Now, compared to normal amplification, normal synthesis, right. So, Sanger sequencing utilizes something called dideoxy termination method. Okay. So, how does this dideoxy termination work? So, it works by having certain modified nucleotides which will stop the reaction. Okay. So, we will illustrate this a bit into this class. So, what we have is the DNA polymerase, right. We need to synthesize the complementary strand. We need primers, right. So, that will bind and then we will get the complementary strand synthesized. We will have pool of unmodified DNTPs. So, these are normal DNTPs that we use in the lab. So, we will have DNTPs means DATPs, DGTP, DCTP and DGTP, plus we have pool of dideoxy NTPs, right. So, these are the dideoxy terminators, right. Once they are incorporated in the synthesized strand, these will block further synthesis. Okay. So, further extension will be blocked, and then there will be some other process that will be done. Okay. Now, what should be the ratio of this unmodified DNTPs and the dideoxy DNTPs? There are specific ratios that we have to follow, again depending on the length of the molecule that we are sequencing, right. So, again there is specific ratio that we follow in this process. Okay. So, just to illustrate this now, so these are the dideoxy NTPs. I have just put them in some schematic, right. So, you have 4 types A, T, G, C, right. So, each of them they are tagged by some fluorescent color, right. So, they have all these different colors, you can see orange, green, yellow and blue for this, right. So, this is again a schematic, right. Of course, these are not the exact colors that are given, but they are different. So, that is the point. So, what we do is that if we have genomic DNA, so what the first step is we denature and fragment the DNA, right. So, we cannot synthesize or sequence the full genome, right. If we are let us say talking about human genome, we cannot directly sequence the full genome in one go. Okay. So, we have to do this in step by step manner. So, what we do is we fragment the DNA, denature and fragment. So, we get single stranded fragments, right. So, we are representing these single stranded fragments here, we get multiple such fragments. Now, we can take one such fragment and we can do the Sanger sequencing. Okay. So, that is how this process works. Okay. So, what we do is, if we imagine we have this strand and we have this primer binding site for which where we know the sequence, right. So, we have to know a part of the sequence to design this primer, and then we synthesize the complementary strand. So, you can see this complementary strand being synthesized. Now, what happens is that when these normal DNTPs are incorporated, the normal extension is going on, you are getting growing strand of DNA. But the moment a modified DNTP, these DDNTPs, these are incorporated, then the synthesis stops. Okay. So, here you can see the right, we have this TTC, and so this is C is a modified one, right, and it will stop the extension. Okay. And similarly, here we have A, so it will stop the extension. Here we have J, right. This will stop the extension. Here we have T, it will stop the extension. Now, this is again by chance, right. In some cases, we'll get these DDNTPs, right, but most of the time we'll get normal NTPs, DNTPs, right. So, this is where the ratio of DNTP and DDNTP plays a very important role. So, this has been optimized through trial and error. Now, for the moment this stops, right, what you can do now is we can take these different fragments and we can try to separate this out and read the signal. Okay. So, you can imagine, right, if you have this fragment of, let us say, 100 base pair, right, you will generate a lot of these fragments of different sizes, right, with normal DNTPs, but at the end one modified NTP or DDNTP. Okay. So, what we do is then we take these samples and run through capillary electrophoresis. This is like your DNA gel, but of course, running through a capillary, right, not just the agarose, it is a different electrophoretic medium. And here you can see 4 samples running 1, 2, 3, 4, right. And what will happen is that you get these fragments generated from each sample, right, of different sizes, they will be separated out based on size. Okay. So, the smaller fragments will run faster through this electrophoretic medium and the bigger fragments will run slower, right. So, if you look from, so if you are running from this direction, right, so after some time what you will see is that the smaller fragments will come down here, right, and the bigger fragments will remain here. Now, what you can do once these fragments are separated, we can now read the signal, the fluorescence signal of each of these fragments, right. So, let us imagine the fluorescence signal here is green, the lowest fragment here is green, the next one is red, so on, right. So, we are reading these signals, and by reading this signal we know what is the base that is present at the end of that fragment, right. So, in this fragment we know the green, so green is T, right. So, what we will get is T, then what we will get is A, red, then we have blue, 1 blue, 2 blue, right. So, 2 C's, then again red, so A, then green T, and so on, right. So, for each lane we can now read this and we can figure out what the sequence of these fragments are, right, because these two neighboring fragments, right, they differ by just size 1, right, 1 nucleotide difference, because there is nothing in between them, right. So, this is how the sequencing is done, right. So, we can now read the sequence from this process. Okay. So, this is the method that was applied actually for doing the human genome sequencing project, right. So, that started quite long time back, and they employed a method called shotgun sequencing, but they used Sanger sequencing to actually sequence the genome. This was a very important project because we thought, right, by looking at the genome sequence we will understand, right, the functions of genes and which genes are responsible, which type of diseases, etc. So, that is why this was a very important project at that point. Okay. So, what they did, so they took genomic DNA from an individual, right. So, they isolated genomic DNA, they did the fragmentation, right, they generated a lot of these fragments. So, you can imagine, right, so human genome size is 3 times 10 to the power 9, so 3 billion bases, and you can get enormous number of fragments, right, and you can start sequencing these fragments, right. So, again some fragments will be present multiple times, right, and you can process each of these fragments to Sanger sequencing. Okay. And once you get these fragments, what you have to do, you have to generate this full assembly of the genome, right. So, the fragments, these are only part of the genome, right. You need to assemble the genome, right. So, our genome is organized in terms of chromosomes, right. So, I have chromosome 1, 2, etc. So, you have to actually build the assembly and say, okay, this is chromosome 1, this is chromosome 2, and so on. As you can imagine, right, we are sequencing these DNA fragments one by one, right. So, in parallel maybe you can do 96 or so at most. So, it will take enormous amount of time to actually sequence all those fragments, okay, and that is what actually happened. So, this was a very time consuming project and it was very expensive, okay, because Sanger sequencing, right, you have to process these through one fragment at a time and you cannot parallelize this process. So, this was completed, the human genome sequencing project was completed in 2003 and it took about 13 years to sequence just one genome, right. So, that is a very long time and the cost was huge. It was US dollars in terms, so it was 3 billion at that time. So, you can imagine how much money actually went into just sequencing one genome. So, you can probably now understand the limitations of Sanger sequencing if you want to do genome sequencing or something like that. Okay. So, this is where the limitations came into picture, right. So, the first limitation of Sanger sequencing is that it is low throughput, right. So, you cannot process too many DNA fragments at once. So, we saw like sequencing is done for one sample, you can maybe get 96 samples at once in the machine, right. So, this is a very low throughput method, you have to generate these fragments in the lab, you have to clone them and then do the sequencing reactions, right, this dideoxy termination reaction for each of these fragments and then run through capillary electrophoresis, right. So, this is a very tedious process and it takes a lot of time. So, when you apply this, as we have just seen, when you apply Sanger sequencing to a very large scale project, genome project, right, this becomes very expensive and it takes a lot of time. Okay. So, this is probably not the way you want to sequence not just human genomes, but also other species right around us, plant genomes, etc., crop genomes, right. So, this is a big limitation of Sanger sequencing project. Okay. So, what people thought about, maybe we can speed up this by using something called microarray. Now, of course, this is not the solution to sequencing new genomes, but you can at least identify mutations, do genotyping and also do some sort of gene expression studies using this microarray technology. Okay. So, what microarray technology does, it uses something called DNA probes which are present on solid surface. So, these DNA probes are designed so that they are complementary to the sequences in a genome. Okay. So, we will discuss this in a moment, right. So, what happens is that you have these probes and you take genomic DNA and you hybridize, and by looking at this hybridization process you can identify whether there are mutations or genetic variation in your sample compared to the reference that is present in the DNA probe. Okay. The limitation is that sequence has to be known, right. So, if you want to synthesize and design these DNA probes, you have to know the sequence of the sample that you are working with, right. So, if you are working with human genome, you have to know the sequence first, only then you can design these probes. So, you see this problem immediately, right. So, you cannot sequence new genomes, right. So, you cannot work with genomes for which you have no sequence data. Okay. Nevertheless, people use this method quite a bit for genotyping and gene expression studies, because this was a throughput method, high throughput method that was available at this time. Okay. So, how does it work? So, we will just illustrate this now. So, as I mentioned, you have a solid surface, okay, and on top of that you have these DNA probes. You have many of them, right. I am just drawing a few, but you have hundred thousands of them. Okay. So, again you can also control, right, how many you can have. So, you can have between hundred thousands to millions. Okay. So, you can have these probes that are complementary to some region of the genome sequence that you are working with. Okay. And what you do now, if you imagine this, this is your sample, you have the cell, right, or some cells like clusters of cells, you isolate DNA, you fragment, right. We have seen this in case of Sanger sequencing, and you label this with some sort of fluorescent label, right. So that you can read the signal. Okay. And then you add this to this array. Okay. So, after labeling you add these DNA fragments to the array, there is hybridization. So, what it means, right. So, if you have complementary sequence, right, you will see formation of double strand, right. So, double stranded DNA molecules will form, and with this formation, right, these labels will be retained on the array, right. So, this is the process of hybridization, right. So, we have these label molecules that are now allowed to bind to these probes, DNA probes here, right, and some of these fragments will form these double stranded DNA molecules on this chip or solid surface. Okay. So, these double stranded molecules will be there and they will carry the labels, right. So, you have this double stranded molecule formation and they have this label L, right. So, these labels can then be read by a detector. Okay. So, there is a detector that will actually read these labels, right, and will identify the signal. Okay. So, how do you actually then use this method to identify mutations? Okay. So, what you will see, let us imagine these two samples, right. So, you have sample 1 and you have sample 2, right, and you want to identify which sample contain some mutations in the DNA. Okay. So, we have these DNA fragments, right, with the labels that we have generated from both samples, and then they are hybridized against this solid surface, right. So, these DNA probes are there. So, you do this hybridization. Now, imagine this sample S2, it has a mutation here, right. So, let us say there is a mutation here, okay, in this position A mutation. Okay. So, what will happen is that since in sample 1, right, these DNA molecules are perfectly complementary to the DNA probes on the array. So, there will be strong hybridization, okay, because there is perfect complementarity. So, this will give very strong signal. Compared to that, because there is a mutation, right, this will disturb this hybridization process, right, because the mutation is not complementary. The part before and after this mutation, they are complementary to the probe, but this mutation is not. So, this will disturb the hybridization process, which means you will get lower signal. Okay. So, you will get higher signal here, right. In case you have stronger hybridization, you will get higher signal. Now, you can also do something else, right. So, in these arrays, when you are designing this DNA probe, you can guess, okay, maybe we will see this mutation, okay, and design probes that are complementary against this mutation. Okay. So, in the array you have both types of probes, one complementary to the wild type sequence and the other set of probes that are complementary to a mutant sequence. Okay. So, you can have these probes, right, wild type probes and mutant probes, right. So, what you will see is that if there is a mutation in the sample, this mutant probe will give you higher signal and the wild type probe will give you lower signal. Okay. So, now if you compare this, so you have these both types of probes in the microarray and you are comparing this S1 and S2, right. S1 will give you higher signal for wild type probe and lower signal for mutant probe, right, because there will be weak hybridization with the mutant probe. And in case of S2, you will see exactly opposite. So, you will get a lower signal for wild type probe, you will have weaker hybridization, and you will see stronger signal or higher signal for mutant probes, because there is very strong hybridization, right. So, in this way you can take a guess and you can design probes that will help you identify gene mutations or genotype, right. So, this is what researchers did with microarray technology. You can also study gene expression using these microarrays. Okay. So, we will illustrate this very briefly. So, again we have this sample 1, right. I am just drawing here again, and they are now bound to these labels that gives us signals. Okay. And in microarray technology, what you will do is you will so for gene expression analysis, you will identify you want to identify or measure expression levels of genes between these two samples, right. So, what we will do is we isolate RNA, then we convert that to cDNA and we hybridize that cDNA to the microarray. Okay. So, these microarrays now designed with oligos, right, with probes that are complementary to mostly cDNA sequences, right, not to against the whole genome, but against only the gene sequences, okay, because the mRNAs will come from the coding sequence or the gene ORFs, right. So, we design these probes so that they only are complementary to the cDNAs. Okay. So, we use the same set of probes against these two samples, right, and we hybridize them, right. This is the hybridization process again. So, once we go for hybridization, what will happen, right. So, let us imagine we are measuring expression level of a single gene X. Okay. So, we have let us say probes against this gene X, right, in the microarray chip, okay, or the array on the array. Okay. So, these probes are present in multiple copies, maybe in hundreds or thousands of copies. So, what will happen is say in this sample 1, right, let us say the gene X is expressed at the level of only 10 copies, okay. And in sample 2 you have only, so here you have only 10 copies of this mRNA produced, and in this sample 2 you will get 100 copies of mRNA produced. Okay. So, in first hybridization you will see only 10 copies. So, and in the probe in the array you have 1000 such probes against this gene X. Okay. Now, if only 10 copies are produced, so not all of these probes will be occupied or hybridized, only a fraction of them will be hybridized. So, the signal will be proportional to this number of copies. In case of sample 2, right, you have higher number of copies, 100 copies. So, a larger fraction of these probes will be hybridized by the sample. Okay. So, you can see now, because there is a larger number of copies, like so what will happen in case of sample 2, you will get stronger signal. Okay. And this way it will tell us, right, okay, in sample 2 you have higher expression of this gene X compared to sample 1, right. So, this can be now repeated for multiple genes, right. So, this is as I said, you can have millions of probes. So, you can do this for 5,000 genes or 10,000 genes, and you can measure the expression levels of these genes, compare expression levels of genes between these two samples. Okay. So, so as you can probably now imagine, right, you need these different types of arrays for doing these two different types of analysis, right. So, you need to have something called SNP array, which will help you identify these mutations or genotypes, and you also need something called expression array that has to be designed for gene expression analysis. Okay. These arrays would have to be organism specific, as you have seen, right. They require these DNA probes, right, for which we need to know the organism sequence, the genome sequence, as well as these sequences are different, right. So, we need these organism specific arrays, and you can also design customized arrays. You can have your DNA molecules that you want to probe, right, you want to see or you want to design as probes, and you can also do this customization, and you can design your own array, and you can print those probes on these chips. Okay. So, with this discussion you can probably now understand the drawbacks of microarray technology. So, it only works with known sequences, you cannot sequence anything that is unknown to you, right. So, you first need to figure out the genome sequence or cDNA sequence before you can use microarray technology. It is not capable of identifying new genes or sequencing new genomes. This is obvious, as you have seen, and this is an indirect method, right. So, we are not directly looking at the sequence of these DNA molecules, we are just looking at the hybridization process, right, which is dependent on a lot of parameters. So, we have not discussed all the details about the conditions in which we do this hybridization. So, these conditions are also very important in determining the hybridization efficiency, right. So, if you have slight modification or fluctuation in this hybridization condition, that can tremendously impact this hybridization efficiency, which means the signals will be affected by this hybridization, right. So, this leads to reproducibility issues. So, if you are doing this experiment on different days, different times, and there are slight fluctuations in your sample preparation, or the salt concentration is slightly higher or lower, this can lead to different signals, and that can lead to reproducibility issues. Okay, so, this actually led to emergence of next generation sequencing technologies. So, what was needed is a direct sequencing method. As you have seen, microarray is an indirect method, and that is actually not good enough. So, we needed a direct sequencing method, and it should be high throughput, accurate and reproducible. There should not be too much error when you are sequencing these DNA molecules. It should be cost effective for large scale project, right. And how do you define this cost effectiveness? Is the target was said that human genome should be sequenced for $1,000 or less. Okay. So, we are there actually already, we have reached that goal, and the goal is to actually take this cost down further. So, what are the benefits of NGS technology? So, NGS technologies is simply scaling up the whole process of sequencing, right, and making it high throughput, right. So, what it does, it can sequence thousands of genes or genomic regions simultaneously instead of just one gene fragment that we have seen in Sanger sequencing. It can sequence unknown genomic fragments, because this is a direct sequencing method, it does not rely on hybridization. It can process large number of samples in a very short period of time. So, you do not have to wait for 10-13 years to get one human genome, you can do this now in 7-10 days. You have more power to detect low frequency variants. So, we will see what these low frequency variants mean when you actually go into the topic. So, these are actually genotypic variations that are present in population and are present in low frequency, or they are rare in the population. And these methods are also cost effective for processing a large number of samples. Okay. So, coming to the application, so NGS technologies have had huge impact on these different fields, genomics, transcriptomics, epigenomics. So, I will just very briefly mention, and of course, you are welcome to go and read a bit more through the references. So, here is some statistics that I will give you. Here is a number of eukaryotes or genomes that have been deposited in these databases. You can see these numbers, right. So, eukaryotes, prokaryotes, viruses, right. So, this has seen tremendous growth in the last few years. So, in last 10-15 years, we are seeing 300 genomes being deposited per month to these databases. Okay. So, this is a direct contribution of next generation sequencing, because it has sped up the process of genome sequencing quite significantly compared to Sanger sequencing. It can be applied in transcriptomics, and we are seeing these applications across many different fields. One of the most important studies of transcriptomics is to look at disease samples and try to understand how gene expression changes between disease and normal samples. So, this can help us give insights into the disease process, right. So, this is very important, and this has been actually done for many diseases now. So, what happens in transcriptomics is we measure expression level of all genes and we sequence all mRNA molecules transcriptome of a cell. Of course, we do not directly sequence mRNA, there are specific processes that we go through, and it allows us to discover novel transcripts, and there are probably transcript variations that can happen from normal to disease samples, and we can identify those. And we can also see something called alternative splicing. You are aware of what is alternative splicing. So, we can look at different spliced variants through this application of NGS in transcriptomics, which was earlier very difficult to do with microarray, because you did not know what kind of splice variants that can appear. We could have taken a guess, again design probes, but that was a very tedious process. So, this has been applied, NGS has been applied for disease states in cancer, and there is a whole bunch of data for different types of cancers where we have this transcriptomic data. So, it is called Cancer Genome Atlas project. So, here is the link where you can actually look into some of these data sets, explore the data sets, and you can also read papers looking at the differences between cancer versus normal samples. This has been applied in epigenomics. So, the epigenome or epigenetics is study of modifications on the DNA without any change in sequences. So, this has been actually a very important field now, where we can apply next generation sequencing technologies to identify these DNA modifications at a very rapid pace, right. So, and again this is very important for many human diseases, and we know, like for example, in cancer, changes in the epigenome are the key important drivers of disease, as well as they can determine whether a therapy would work or not. So, here is a reference which you can study, right. So, we are talking about the epigenome studies across human diseases. And finally, these methods have been applied in something called metagenomics. So, again this is a study of microbial community. So, for example, we have something called soil microbiome or gut microbiome. So, we have these microbial species that are living inside our body, and we can study this, and this has very important clinical implications. For example, for disease detection, which kind of treatment that you can specify, or if you can develop novel therapy that can prevent certain type of diseases, right. So, probiotics are a direct outcome of this. You can also use this metagenomic study for environmental bioremediation, where you can design communities of microbes to clean up certain pollutants such as plastics or hydrocarbons, etc. Now, NGS technologies, so through this application of genomics and transcriptomics, they have built something called precision medicine, right. So, what you can do is we can now study patients in much more detailed, right, rather than just the symptoms or some traditional diagnostics. We can now actually study the genomes, the genomic changes, what are the transcriptomic changes, right, and we understand, right, even two persons with the same disease may not respond to the same therapy, right. So, not all individuals will respond to the same therapy, and this does not work, and this has been seen a lot in cancer. And this is because there is differences in genotypes in two individuals, also how the disease is progressing, right, which we can now study through analysis of NGS data, and we can then design specific therapy for specific genetic basis of disease. So, this has been termed as precision medicine, right. So, we do not give same treatment to all patients with the same disease. We can identify subtypes of the genetic basis, and we can specify specific treatment for each patient. Now, again, with all the data that are coming in from all these fields, we can also devise machine learning approaches, right. So, this is where the data science field also merges with biotechnology and biological sciences, right. So, we have data sets in genomics, transcriptomics as well as epigenomics, and we can take these data and apply machine learning models to utilize for classification and prediction tasks. This could be for example, disease subtype classification. I have given you an example here, something like cancer subtype classification from transcriptome data. So, there are lot of advantages, lot of benefits that we have seen from NGS technologies, but there is also some limitations which we probably can address, right. So, one is it is not as cost effective for processing a small number of samples. Now, this will probably will not be a major problem as the price or the cost of these technologies are going down. Now, the second problem is the time, it is very time consuming, right. If you are processing a small number of samples, but then you can probably go through the Sanger sequencing route. And in addition, with the newer advancements, we are actually making these steps much simpler, the library preparation, etc., so that we take less time. And these technologies have benefited so much that we can now take these single cell approaches, where we can actually apply this genomics, transcriptomics, and epigenomics to the study of single cells instead of full organism or a whole tissue, and this has actually has given us tremendous insight into the biology or workings of these biological systems. These are the references that we have followed for this class. To conclude, so next generation sequencing technologies have provided really high throughput, accurate and cost effective solutions for direct sequencing. We have seen they have actually accelerated the pace of new genome sequencing to a great degree, and finally, we have talked about the applications across diverse fields and how they have provided tremendous benefits in clinical settings. Thank you.